The tool Surat Setup and QC outputs some quality control plots in a PDF file. But you can also use Scater QC tool to get another PDF with several QC plots. The tool is very simple. It takes a Surat object as an input, and there are no parameters. Inside the tool, the Surat object is transformed into Scater format, and several QC metrics are computed. The tool then plots some of those. Note that you can open the PDF in a new tab. This makes viewing it much nicer. The output plots depend a bit on which data fields are available in the Surat object used as an input. If the cell cycle scores are available, then those are used in the plotting. Let's take a look at the plots. We are looking at the basic Surat tool demo session with one sample. The first Surat object resulting from the setup tool is used as an input for this particular case. The first plot shows the features with the 50 highest average expression across all cells, along with their expression in each individual cell. Y-axis lists the top genes, and X-axis shows the percentage of total counts. Each small tick or bar represents a cell. The color of the tick represents the total number of features in that cell. The dot in each line shows the average expression. In this plot, you can check which genes are the top 50 most expressed and detect possibly overabundant genes. For example, you can see if there are mitochondrial genes, starting with MT, that you might want to remove from the analysis. The second plot shows the relative proportion of the library size that is accounted for by the 1,000 most highly expressed features for each cell. Y-axis is the cumulative proportion, and X-axis the number of the top expressors included. Each cell is represented as a line. This plot can be used to look for differences in expression distributions between samples. On the third page of the PDF file, several quality control measures are plotted against each other. For example, on the bottom right, you can see the mean expression versus the number of cells with detection. Again, a dot is a cell, and the X-axis tells you how many cells there are with, for example, 2,500 genes detected, and what is the mean expression in those cells. The different quality control measures are explained in the Scater documentation if you look at the Calculate QC Matrix function. Here they list the different measures and their meanings. On pages 4 and 5 of the PDF file, we can see the first 4 principal components plotted against each other. The percentage of the variability of the data explained by each PC is also reported. You can see it here. The dots representing the cells are colored based on the mitochondrial percentage and the total number of features. Here, you might be able to spot if some of the PCs would be clearly driven by mitogenes. And you can also see that the number of features plays a role in many of the dimensions. The rest of the pages are explanatory plots. With them, we can investigate the relative importance of different explanatory factors. In the first 2 plots, a density plot is created for each variable, showing the distribution of R² across all genes. Scater calculates the R² for a factor or variable by fitting a line for each gene against the factor or variable. R² is the proportion of variance for dependent variable explained by an independent variable. We can also identify principal components that correlate strongly to certain quality control factor or metadata values. The two last plots represent these. In both cases, some selected factors are plotted. If there are cell cycle scores present in the surrogate object given as an input, the values for the phase, G2M score and S score are also plotted.